Welcome to Lecture 11, where we will go over factors that affect animal diversity. Animals are considered to be highly diverse. There are some key characteristics that make them very efficient at consuming other animals. Most animals are considered to be heterotrophs, which means that they need to obtain their energy and nutrients through the consumption of other organisms. As a result, they have specialized tissues that have formed from embryonic germ layers that allow them to carry out this function. Many of these animals have an efficient digestive system that allows them to extract nutrients from the material that they consume. In addition, they have various types of nerve cells as well as muscle cells that will help to control and also regulate this process. In addition, many animals can move throughout their environment in order to move, detect, and capture potential prey items. Though animals are considered to be highly diverse, they have many traits in common. These include the fact that animals are multicellular organisms, though these cells are eukaryotic based in nature. These cells can be organized into various tissues that carry out specialized functions, and these tissues can also be further organized into organs as well as organ systems to help the animal survive and thrive and adjust to its surroundings. Animals are also considered to be heterotrophic. This means they have to obtain their nutrients from the consumption of other organisms or from the consumption of non-living organic material. They do not make their own organic compounds for energy, as we've seen in autotrophs or plants. Many animals reproduce sexually through the production of sperm and eggs. The sperm and egg are produced through the process of meiosis. Remember, this is a reproductive strategy that greatly increases the genetic diversity of the offspring. In addition, animals are present in primarily the diploid or 2N stage. This diploid stage will dominate the life cycle, while the haploid stage is typically represented in the production of gametes that are haploid. Animals contain small flagellated sperm that are haploid. The sperm will fertilize the ovum, which is also considered haploid. Once this fertilization process occurs, this leads to the formation of a diploid or 2N zygote. This zygote will then undergo a process called cleavage. This means that the zygote, which is originally one cell, will undergo repeated mitotic divisions. As this occurs, this leads to the formation of the blastula. The blastula then forms a hollow and ball-like structure. This blastula will then undergo a process called gastrulation or invagination. This will then form a gastrula. Once this process occurs, the invagination occurs, we now have the differentiation of two different tissues called the endoderm or inner tissue, as well as the ectoderm or outer layer of cells. Embryogenesis will continue along these lines of repeated mitotic divisions as well as repeated differentiation of cells to form various types of tissue. Embryogenesis can involve many different larval stages, or it can involve a complete metamorphosis from a larvae to an adult. Though the adult morphology of animals varies greatly, the genes that actually control this process are considered to be highly conserved. An example of this is the Hox genes, which we will take a look at various points in this lecture. This slide demonstrates the early process in embryogenesis. Note that we have a one-celled zygote. Remember that the zygote results from the fusion of the sperm and the egg. Thus, it is considered to be diploid, or has two sets of chromosomes. One set would have originated from the sperm, and one set would have originated from the egg. Once a zygote forms, it rapidly undergoes the process of cleavage to form the blastula. As the cells divide due to mitosis, they will be organized into the blastula stage. This means that there will be an inner cavity that can be termed the blastocele, and that cavity is surrounded by a layer of cells. This will then undergo gastrulation to form a gastrula. This means that there will be an invagination that we can see in the diagram. This invagination will lead to the formation of two different cell layers. The inner cell layer, which is termed the endoderm, the outer cell layer, which is termed the ectoderm, and the cavities that reside within these layers. The blastocele refers to the cavity between the ectoderm and endoderm, while the archenteron refers to the cavity that resides within the endoderm. Also note the presence of a blastopore. This can then differentiate into the mouth or anus depending on the animal concerned. Though many animal species have been described, there are still many species that yet remain undiscovered. Animals have diversified greatly over the last billion years. Various lines of evidence in the fossil record as well as molecular clock studies suggested that sponge originated approximately around 700 million years ago. However, more recent DNA studies suggest that the common ancestor that existed prior to this, that gave rise to the sponge and animal lineages, originated approximately 770 million years ago. This slide demonstrates the phylogenetic relationship between choanoflagellates, sponges, and other animals. If you recall, choanoflagellates are individual unicellular eukaryotic organisms. They bear a striking resemblance to the collar cells in the multicellular sponges. Also recall that sponges are considered to be a sister taxa to all other animals. There are many lines of morphological as well as molecular evidence that identify choanoflagellates as the closest living relative to modern-day animals. It's hypothesized that the common ancestor would have been a suspension feeder very similar to what we see in present-day choanoflagellates. Also keep in mind there are some key adaptations that are required in order to progress from a unicellular organism to a multicellular form. These include the ability to adhere to other cell types, and as well the ability to communicate between different cells. One line of support for this relationship involves the examination of cadherin proteins. If you recall, cadherin proteins are important in maintaining attachment sites between adjacent cells. If you take a look at the relationship within the phylogenetic tree, you can see that there are both some regions of cadherin proteins that are highly conserved, as well there is some divergence or novel encoding regions, such as the CCD region or cadherin coding domain. This domain is only found in animals, representing the divergence from a common ancestor. The Neoproterozoic era occurred approximately 1 billion to 540 million years ago. This era involves the origin of modern-day animals. It also includes the Ediacaran biota, which is commonly found in fossil records obtained from the continent that we now know as Australia. Within the fossil record, there are fossils from many soft-bodied multicellular eukaryotes, such as mollusks, sponges, cnidarians, and their relatives. There is also evidence of predation, confirming the heterotrophic nature of these organisms. This can be seen in the borehole present in the fossil below. The Paleozoic era occurred 540 to 250 million years ago. This era includes the Cambrian explosion, which is a distinct radiation event that contains all major extant animal phyla. This means that all modern-day living animals can be traced to some sort of origin within this time. This includes arthropods, chordates, echinoderms or starfish, and their relatives. During this time, we also see a distinct relationship between the evolution of predators and prey species. This is typically called the predator-prey arms race. Predators evolved different prey-catching structures, while prey evolved many defensive structures to evade predation. Also, during this time, these fossils contain many different animals with hard mineralized skeletons. We see an overall decline in the soft-bodied eukaryotic and multicellular organisms that we saw in the previous era. The overall decline in Ediacaran biota is due to many reasons, including their susceptibility to predation given their soft-bodied forms, an increase in atmospheric oxygen that occurred during the time which allowed for larger animals to thrive, and the genetic evolution in various types of developmental genes, such as the Hox genes and microRNAs. This allowed for more complex body forms to evolve. The following periods, which include the Ordovician, Silurian, and Devonian periods, were punctuated with many different extinction events, which were then followed by explosion events. We can see this in the vertebrate fishes when they radiated, and became the top predators in marine systems. And we also see explosion events that follow major evolutionary events, such as the movement of arthropods onto land, then followed by vertebrates and their diversity. The Mesozoic era occurred 250 to 66 million years ago. During this time, various types of habitats in both the marine and terrestrial environments evolved. Some reptiles returned to an aquatic environment, whereas large and small dinosaurs remained in the terrestrial environment. During this time, we also see the evolution of flight in tetrapods, and the first mammals and angiosperms appeared. This also led to a diversification in both the angiosperms and the insects as a mechanism of coevolution. The Cenozoic era occurs from 66 million years to present day. During this time, we have the mass extinction event that led to the extinction of dinosaurs and as well as many marine reptiles. As a result, we see a radiation event in mammalian species. This has led to the dominance of large mammalian herbivores as well as predators. A gradual climate cooling that occurred led to a species shift in adaptations to adapt to new climates, habitats, as well as ecosystems.